Imagine an environment journey to the brain's resting state where neurons and glial cells work together like a well-orchestrated team. At the core of this story is the incredible process of three-color tracing. Think of it as giving each neuron its own unique badge so we can follow through the paths through the connection. This helps us understand how signals travel along axons with conduction velocities that keep everything running smoothly. Now picture insulin resistance sneaking into the brain's metabolic neighborhood, making it harder for neurons to access their favorite fuel, glucose, via insulin-dependent glucose transporters. When glucose isn't available, neurons can switch gears and tap into ketones transported by ketone transporters. These ketone bodies, especially ketone monoesters, become alternative energy sources supporting mitochondrial function and helping stave off metabolic stress. But sometimes things go awry. Lipid transport proteins and neuronal insulin pathways can falter, leading to glucose hypometabolism, a kind of energy drought that can cause neuronal atrophy and contribute to cognitive decline. This energy crisis often comes about mitochondrial dysfunction and microglial activation, which can trigger inflammation and compromise the blood-brain barrier, allowing immune cells to enter and potentially cause further damage. As these processes unfold, neural proteins like beta amyloid and tau proteins start to accumulate, disrupting neuronal signaling and connectivity. The hippocampus, our memory hub, and regions like the putamen and supplementary motor areas are affected, impairing not just memory but also motor function such as motor coupling. This can lead to increased motor variability and challenges in stimulus encoding, especially in auditory motor sequences involving the geniculate nucleus, thalamus, and supracortical. On the cortical level, structures like the primary visual cortex and lateral extrastriate areas engage in complex processing, gain control, surround suppression, and orientation selectivity that shape our perception. The parietal, limbic, and temporal cortices work together through dorsal and ventral streams, each with distinct cytoarchitecture and electrophysiology, form an intrinsic mapping system that underpins our understanding of space, emotion, and memory. All these neuronal activities are influenced by blood system. Neuroendocrine and autonomic nervous system regulate human sensory and motor activity, while vascular processes ensure nutrient delivery and waste removal. Inflammation and cellular synthesis, including the senescence associated secretory phenotype, can disrupt brain homeostasis, leading to further cellular decline. Scientists use techniques like anterograde tracing to map these neural pathways, revealing how the brain structure and function are intricately linked. It's a delicate balance. Metabolic, immune, and vascular processes all converge to support neuronal health, connectivity, and electrophysiological functioning. When this balance tips, it can lead to neurodegeneration. But understanding these pathways gives us hope for interventions that keep our brains healthy and vibrant well into the future.